want to stop. Okay, you feel like you're overwhelmed, you're getting stressed, you want to stop. Drink a lot of water. Okay, and then do this breathing exercise. You breathe in for 4 counts, hold for 7 counts, and breathe out for 8 counts. Okay, so you do that for 5 cycles, you will mentally calm down, and then you'll be in a better place to learn. Also, I would suggest using this free app called Focus Keeper. So this Focus Keeper app lets you study for 25 minutes, take a 5-minute break, study for another 25 minutes, take a 5-minute break, and you keep going like that for 4 sessions. Then you get a 15-minute break. Okay. So during those 5-minute breaks, you're not supposed to be looking at your cell phone, anything electronic. Okay. Not your computer, not going on to another website, nothing. You're just supposed to chill out. You can look through the window, enjoy the birds, enjoy the weather, enjoy your pet, right? But not look at the computer. Okay. So if you do that, the thing is, your brain is like a muscle. Okay, it can work for some time and then it needs to relax to recuperate. Then if you study that way using that method, then you retain the information better. You don't have to kill yourself studying. At the end of a 2-hour or 3-hour study session, you're not mentally exhausted. Okay, your brain muscle is weak. Yes, I agree. No, I don't. Okay, you just need, it's like you have to kind of use the technique, and it's supposed to work with like what is that line of study? Supply chain management. Okay, they use the technology, it seems, and it works really well. So you have to do that for your brain. Okay, study, take a 5-minute break, study, 5-minute break, and go on. We do that for grants. You know, I'm getting older, so when I write grants, I get like really exhausted because we pull in all-nighters and stuff like that. But if I use this technique, it is awesome. I'm fresh as a daisy after the whole thing is done. Okay, so let's move on. So the first thing to learn about in histology is connective tissue. Okay, so let's pull up some connective tissue sections and I will share my screen. Okay, so with connective tissue, you should know the different types of connective tissue, basically. Okay, so you look at dense, regular connective tissue, dense, irregular, loose, irregular connective tissue. Okay, so you have to know what they are, like look at pictures, okay, and be able to identify where all these different types of connective tissue are. Okay, so get all these connective tissue fibers and be able to identify what is loose, like right here, and this would be
Dense.
Dense.
dense what?
Dense regular.
Dense regular connective tissue proper.
Uh, looks like dense regular, doesn't it?
Mm-hmm.
Yep. Okay, so tendons and ligaments. Maybe there is a tendon or something. Oh, there is no tendon attached to the ear. This is ear because this is
Elastic.
yeah, elastic cartilage. Okay, so this is the ear. So this is what? What would you call this?
Dense irregular.
Irregular.
Dense irregular. Okay, and then this would be
Dense.
this
Irregular.
loose irregular, right? So you have to be able to identify all these different types of connective tissue. Then what is the main type of fiber that is present in most of the body?
Collagen.
Collagen type 1.
Type 1 collagen. Okay. So where do you find type 2 collagen?
Hyaline cartilage and fibrocartilage. Or no, elastic.
Elastic cartilage. You won't see it in fibrocartilage because it's mostly
Type 1.
1 collagen. Okay. So type 2 is really fine collagen fibers. What is type 3?
Reticular.
Who secretes those?
Only reticular cells.
Yes. Okay. But type 1 is secreted by?
Fibroblasts.
Yes. Okay. And type 4 is where?
Basement membrane.
The basement membrane. So if you look at the epithelium, I want you to this move there. Okay, give me 1 second. Let's move this. So here is an epithelium, right? So when we are in this epithelium, you will tell me what type of epithelium this is.
Stratified
Stratified
squamous.
squamous.
Yes, and then what else?
Cornified.
Yes, stratified squamous cornified. So if we go down to the very bottom, like down here, okay, under the basal cells, there is a membrane on which this epithelium is actually sitting, okay, and it's not like a sheet, it's like a mesh, okay, so we call it a lattice, okay, so that is what this is. So type 4 collagen makes up this lattice, okay, so type 4 collagen is present in the basement membrane, okay. Where would you find elastic fibers?
Tendons.
In tendons,
In the vessels.
in blood vessels, right? Like especially in uh, what? What kind of
The
blood vessel?
Arteries.
Artery. What artery can you think of?
Elastic artery.
Elastic arteries.
Elastic arteries, right? Like the aorta and the pulmonary artery. Those are all elastic arteries, and they'll have lots of elastic fibers. Where else can you see elastic fibers?
Elastic cartilage.
Elastic cartilage, awesome. Where else?
Muscle.
Not really. In
Okay.
organs, it can be intermixed with muscle in organs that need elasticity. Okay, so like the lung, for example, or the intestines, you will see elastic fibers intermixed with the muscle or connective tissue there. Okay, so wherever you need elasticity, you're going to see these elastic fibers. Okay, so that is that. And then we have to go to connective tissue cells, right? So we have fibroblasts, we have reticular cells. Who else are we missing? There is another type of cell right here. Who is this?
Adipose.
Adipocytes.
These are all adipocytes. So adipose tissue is also another component of connective tissue. Okay. So now let's move on to the cartilage. Okay. So who is this?
Hyaline cartilage.
Oh no, sorry, I was wrong.
Fibrocartilage.
Fibrocartilage. Awesome. Okay. Hey, by now you should be able to look at the slide, not even open it up, okay, and be able to identify what it is. That's what Dr. MK used to do when we were in school. You know, bad boy. He wouldn't even look at it under the microscope. He would just look at the slide and tell you what it was, because we had only like 30, 40 slides and he had them all memorized. So if you put it under the microscope, he won't know what it was. But if you give him the slide, he will tell you what tissue.
Damn.
So you should be able to do that on the slide set. But here, look at these. So who are these cells?
Chondrocytes.
Chondrocytes. Okay, and they occupy spaces called
Lacunae.
lacunae. Okay, so this is typical fibrocartilage. Where would you find fibrocartilage?
Tendons and ligaments.
Where exactly in tendons and ligaments?
The attachments to bone.
I thought
Yes, when
was
they're
it area
going and attaching to bone, that's when you'll find it. Okay, because it's a transitional tissue. Do you see it? It goes from dense, regular collagenous connective tissue to bone. Okay, so it cannot just go jump from one type of tissue to another, right? So there has to be like an interconnecting, bridging tissue in between, and that is what this fibrocartilage is. Okay, and here is another type of cartilage. Who is this one?
That's
hyaline?
hyaline.
That's hyaline cartilage. Awesome. So hyaline cartilage has an outer layer called the
Perichondrium.
perichondrium. Does the fibrocartilage have a perichondrium?
No.
It doesn't, because there's lots of collagen fibers intermixed in that whole tissue. You don't necessarily have to have like a perichondrium protecting it. Okay. And okay, so again, we have these clusters of chondrocytes called
Isogenous nests or something?
isogenous nests, and the matrix around them is called
Territorial space.
territorial matrix, and the
Can you
matrix
repeat
in
what those nests are called?
isogenous nests or cell nests, and the matrix surrounding them, right around them, is called
Territorial matrix.
territorial matrix, and the matrix in between is called
Interterritorial.
interterritorial matrix. Awesome. Okay, so who are these guys on the very outside?
Chondroclast.
Chondrocytes.
No, these are chondrocytes, right?
So those are chondroblasts on the outside.
They're all chondroblasts. Okay, these are all immature chondrocytes. So as they mature, they're going to secrete this matrix, and then they are going to be trapped in the matrix itself, and then they divide, and that's how the isogenous nests come into existence. Okay, so what is in this matrix?
Hyaluronic acid and gags.
Yes, hyaluronic acid and gags. Awesome. And what else?
Proteoglycans.
Proteoglycans. Okay, what else?
Type
2
2 collagen.
Type 2 collagen fibers. Okay, so that is also present in this matrix. Okay, but you can't see the fibers because they are very thin fibers. Okay, so this is hyaline cartilage. So who is this?
Elastic cartilage.
Elastic cartilage. Awesome. So this is identical to hyaline cartilage except for the presence of these extra elastic fibers. Okay, so you will see it in places where you need more flexibility, like your external ear, your nasal septum, you know, the tip of your nose, and all of those. You will see elastic cartilage. Okay, so now let's move on to bone. Okay, so let me get a good picture of bone. Okay, let's look at this. So who is this from? Do we know?
Isn't that a mouse fetus? A baby's head.
That's the head. You're right, this is from a fetus. Okay, so what type of connective tissue is present in here?
Mesenchymal.
Mesenchymal. Okay, mesenchymal connective tissue is what we have here. Okay, so this will be present. So this is immature bone, right? So what do we call immature bone?
Woven bone.
Woven bone. Okay, very good. So if it's blue like this, that means it is getting mineralized, but this is trichrome. So all this will be collagen. Aha, good examples of different types of cells in bone. So what are the cells on the very outside?
Osteoblasts,
Osteoblasts. The ones that are trapped inside?
osteocytes.
Osteocytes. And who are these big guys?
Osteoclasts.
Yes. Okay. So these are the ones that are involved in dissolving bone and remodeling it. Okay. So very good. Good examples right here. Okay, let's move on to another section of bone. Okay. So what do we have? Let me go 10x. Okay. So what is here on the very outside of bone?
Periosteum,
Periosteum is the membrane, right? It has two layers.
the
Do you know what those two layers are?
outer fibrous and inner cellular layer.
Yes, outer fibrous and inner cellular layer. Okay, so all this is solid bone. Okay, this is part of a long bone, obviously. So this would be the what part of the bone?
Diaphysis,
The diaphysis, but if you're taking a histological section, you would
So
call
the
this the
cortical bone?
cortex. Okay, the cortex of the bone, and this would be the
Yeah, or compact, right? Dr.
Medulla.
Sheba?
the medulla. Yes,
It's also known as compact bone, right?
yes, yes, that is true. This is
That
also
was in the chat, so I just wanted to ask.
okay. Okay, so this is called compact bone. Also, what is the opposite of compact bone?
Medulla. Medullary.
This
Spongy?
spongy bone, yes, okay, or trabecular bone, whatever you want to call it, okay. So that is the opposite. So this one is compact, that one is more spongy, okay. So what is this right here?
The physis.
The physis, right? So there are certain changes that are happening. I don't know what I did right here. Let me yo, the whole thing disappeared on me. Let's move back. Okay, so let's go to the physis. Okay, here is actually, you can see different zones, right? You have to tell me what these zones are. Okay, what is the zone right there?
resting,
Resting, and this would be
Proliferation.
proliferating zone. This would be
hypertrophy,
hypertrophy, and this would be
calcification.
calcification or mineralization. Okay, so let's move down into like this space right here. Okay, so you have periosteum on the outside, right? Outer fibrous, inner cellular. What is the lining in here called?
Endosteum,
Endosteum. Okay, and all this is bone marrow. So the inner aspect of the bone, bony spicules, is going to be lined by endosteum. Okay, so there are some weird looking cells here. Let's go closer. Who are these?
macrophages,
Think again.
Megakaryocytes.
Megakaryocytes. Okay, and these give rise to
platelets,
platelets. Very good. Okay, and these are all mature, immature blood cells. We have different kinds. Like if you look at lymphocytes, the bone marrow gives rise to what kind of lymphocyte?
B cells,
B cells.
T cells. Very good. Okay, so let's move. Okay. So that is as far as bone is concerned. But, okay, let's look at all the different cell types in bone. So we saw the osteocytes, right? They are occupying spaces called
Lacuna.
lacuna. Okay, so let me go and pull up another image so I can ask you some questions.
Dr. Sheba, when you like scan through the images, can you pause for like an extra half a second when you're showing us like with the mouse what you're looking at? Because your
Okay.
voice is on time, but the image is just lagged a little bit.
Okay, no problem. I will do that. I will go a little slowly because I'm rushing through this. I shouldn't. Lessons. No. Open. Maybe this is also bad. How do I show you those pictures? Where do I have those pictures? Let me see quickly. Okay, tell me about bone. What are the different cell types that are present in bone?
You have osteocytes, osteoblasts, and osteoclasts.
Osteoclasts, those are the three main types. Okay, so the osteocytes are arranged in rings called
Osteons.
the whole system is the osteon, right? So that is why I need that image. Give me one.
Is it lamella?
The lamellae, right? Give me 1 second. Where is... I thought I had. Oh, there it is. My mouse won't work. Okay, let's go. Let's see. So they are arranged in lamellae, right? So there are different kinds of lamellae. You would agree, right? So the ones on the very outside of bone, in cortical bone, on the very outside, right under the periosteum, what do you call that lamellae?
Circumference something.
Outer circumferential lamellae.
Something to do with going around something.
Yes. Oh, I know where I have that picture. Give me 1 second. I save all these pictures somewhere and I can't find it when I need it. Because I took a picture of this model that we have in the wet lab. Did you guys get a chance to see those models that we have?
Mm-hmm.
I put it over here somewhere else. Pictures. Oh, there it is. Okay. Are you still seeing my...
Yes.
Okay, can you see this model now?
Yeah.
Okay, so here is bone. Okay, so you can see some lamellae there, right, on the very outside. And there are osteocytes sitting in the lacunae. Okay, so this is the outer circumferential lamellae. Okay, and then if you go down, let me move this. So where the endosteum is, okay, there are some more circumferential lamellae right here. These are called the inner circumferential lamellae. Okay, and then in between you have these circles, right? So each one from here to here is called a
Osteoid.
osteon. Okay, what is osteoid?
Osteoid is the background. The
The matrix,
Extracellular.
right? the organic matrix of bone is called osteoid. Okay, so this is an osteon. So this osteon has the central space called the
Central canal.
central canal. It goes by a specific name.
Cavern.
Come. You're close.
I know. It starts with an H. I know that. It's like
Yeah.
Haversham.
Aversion canal.
Haversian canal. Okay, this is the Haversian canal. You can see blood vessels running through it. Okay, blood vessels and nerves run through the Haversian canal. And so the Haversian canal with the circumferential, the lamellae around it, okay, with the osteocytes is called an osteon. So there is one osteon here, another osteon here, another osteon here, another osteon here, and so on. Okay, and as the bone grows, new osteons pop up. Okay, so as a result, when the new osteons are growing, the old ones kind of move into the background. Can you see the remnants of the old ones right here?
Mm-hmm.
So these are called interstitial lamellae. Okay, there is outer circumferential lamellae, inner circumferential lamellae, the lamellae associated with each osteon, and then you have the interstitial lamellae. Okay, so the osteocytes are residing in spaces called
Erosion lacunae.
the erosion lacunae are the spaces occupied by osteoclasts. Okay, they're also called Hoship's lacunae, but you don't have to worry about that name. Okay, so there are these lacunae are just regular lacunae that are occupied by osteocytes. So when the osteocytes are here, they are living cells, right? They need to get their nutrition and they need to connect with other osteocytes right next to them. How do they do that?
Canaliculi.
Through the canaliculi. Okay, we don't see the canaliculi here, but let me show you another picture of a good bone section. If I can, I keep all these pictures, but then let's open this up. Open this up. There we go. Okay, so this is how ground bone looks like. Okay, you have the Haversian canal in the middle and all these osteocytes occupying the lacunae, okay, and the lacunae communicating with each other through these fine canaliculi. Okay, so the Haversian canal runs in the middle of the osteon, right? So what if the blood vessel has to branch out, okay, and move on to another osteon? How does it do that?
The perforating canals.
The perforating canals or the Workman's canals are the ones that help the bone do that. Oh, I don't have perforating canals in here anywhere. But you know how it looks like, right? Oh, I have another picture here that I can show you. There we go. Okay, so here's a Haversian canal, right? These are all perforating canals. Do you see it?
Mm-hmm.
Okay, so you have to look at this one. It's very pretty. So this is how it looks in cross section, and this is how bone looks in longitudinal section. So most of the sections that we have in your slide set are longitudinal sections. We don't have anything of ground bone, so which is all right. So let's go back to our class and pull up bone. So what other question do I have to ask with the bone? So we looked at the edges, the proliferation, the cortical bone, and all the good stuff, the medulla and everything. Okay, we are done with bone. Let's move on to muscle. Okay. Who is this muscle right here?
Skeletal muscle.
This is skeletal muscle. So how would you identify skeletal muscle?
It's striated and the nuclei are going to be peripheral, not central.
True. Okay. So the muscle cell like the cells themselves are larger. Okay, in cross section, nuclei are always located in the periphery. Okay. Let's move on to connective tissue, cartilage. Where is muscle? Okay, so let's look at some more examples of different kinds of muscle. Hey, you know what? We forgot about epithelium altogether. What happened? Let me go. Mm, okay. Okay. So let's do some epithelium also while we are on this slide. Okay. What is the epithelium that you see here?
Simple squamous.
Simple squamous epithelium. Okay. So this is covering actually the spermatic cord. So what would you call this whole region from here to here? This is in the abdominal cavity.
Mesothelium.
The mesothelium, but uh, the mesothelium is just the epithelium, right? The simple squamous epithelium is called mesothelium. But this whole thing, the epithelium with this underlying connective tissue,
Capsule?
no,
Lamina epithelialis.
no, 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 no,
Propria. Anium.
no, 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 no.
Visceral
The
peritoneum.
outer covering of an organ.
Viscera, the visceral serosa.
Serosa, okay? So that is what it is, okay? This is serosa. So serosa is made up of your mesothelium with underlying connective tissue, okay? Very good. So, okay, let me see if I can get another example. Okay, let's go to this slide. Let's go closer. Okay, let's go little bit. Okay, so there it is. Okay, what is this again?
The simple squamous epithelium.
Epithelium. So this with the underlying connective tissue here would be called
Serosa.
the serosa. Okay, so there are serous membranes that are covering the outside of all organs, all visceral organs. Okay, but what is adventitia?
It's when it's the outside lining of it, but it doesn't have the epithelial lining it because it's in contact with something else.
Yes, it's in
It's
contact.
just the connective tissue, right?
It's just connective tissue that's on the outside. Okay, it doesn't have a clean epithelium, mesothelium covering it because it's not in a body cavity. Okay, it's either running through the neck or that organ is trying to exit the body, you know, in the rectal area where it's like going to be surrounded by muscles. So you won't have a serosal layer covering it. Okay, you'll have a layer of adventitia covering it. Okay, at that time. So this is simple squamous epithelium called mesothelium here, right? So, okay, let's go down further. Good example. Let me find a good example. Okay, what is this epithelium?
Endothelium.
This is endothelium, simple squamous epithelium, but we call it endothelium, right? So this would be what kind of a vessel?
Artery or vein.
Artery.
An artery. This would be what?
Arterial.
Yes, awesome. So that would be a
Venule.
venule. Very good. Okay, and if this space right here would be
Lymphatic vessel.
lymphatic vessels. Awesome, very good. So these are all different types of blood vessels that you can see in this somatic cord right here, because this is the pampaniform plexus, we call it. And this tissue right here would be
Adipose.
adipose tissue. Very good. Okay. So now let's move on to one more structure. I'm going to go closer. Okay. What type of epithelium is this?
Simple columnar.
Simple columnar epithelium. Very good. Okay.
With goblet cells.
With goblet cells. Awesome. So you're able to see a surface adaptation here. What is this?
Microvilli.
Microvilli. So are the microvilli motile or non-motile?
Motile.
No, they don't move. Microvilli do not move. Who moves?
Cilia.
Villi.
Cilia. And what are the structures capable of movement?
Stereocilia.
Stereocilia does not move. It has an absorptive function. Therefore, increasing absorption.
Flagella.
Flagella. Okay. So the flagellum moves. Cilia move. Okay. But these microvilli, they don't move. Okay. It's an adaptation to increase surface area.
Absorption.
What it is. Okay. So here, when we are in this area, what are these structures?
Villi.
Villi, right? Okay, we'll come to this back when we are in the digestive tract. Okay, so let's look at this structure here. I'm going to go down closer. You have to tell me what epithelium this is.
Epithelium or epithelium in transition or transitional epithelium.
Transitional, these are two different things. Epithelium in transition and transitional epithelium.
One in transition is related to respiratory.
Yes. So you don't want to use the wrong term. Okay. So this is transitional epithelium because we are, you know, where we are in the urinary bladder. Okay. So this is just transitional epithelium. Any questions? So this is the transitional epithelium. You have to look at several different examples of transitional epithelium because sometimes it's nice like this, okay, but sometimes it can be like pulled apart. Let me see. Sometimes it's really cool, like here. Can you see it? This is from a horse. Beautiful transitional epithelium. Okay. Different examples is all I'm trying to show. So if you look at the kidney right here, it's another piece of transitional epithelium over here. See how different they look, you know? So you have to look at different examples of transitional epithelium and be comfortable with it, okay? Okay, so when we are here, I can ask you questions on some more epithelium. Okay, what is this epithelium right here?
Simple cuboidal.
Cuboidal epithelium. This epithelium here.
Squamous.
Simple squamous epithelium, simple cuboidal epithelium. Okay. What other epithelium do we have? Let me go back. Connective tissue. Let's go look at this epithelium here. Beautiful. Whoa. Who is this?
Pseudostratified columnar with goblet cells.
Yes. Awesome. Okay. Ciliated. You can see the cilia here. Right? Beautiful. So this is respiratory epithelium. Okay. So we went through the different types of epithelia. Where did I have my cheat sheet? We looked at bone, we looked at cartilage, muscle now. Okay. So let's go a little lower mag to this region here. Chin, 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 chin. Okay, what is this muscle?
Smooth muscle.
Smooth muscle. Okay. How would you describe smooth muscle?
Non-striated, centrally located nuclei.
And spindle shaped muscle cells. Okay. They are shorter, smaller cells. Okay. And they are non-striated. So that's why we call them smooth. Okay. So who is, give me one more second. Okay. Okay, this is just in cross section, right? Look at this muscle here in cross section. And look at it here in longitudinal section. Who is this?
Cardiac.
Cardiac muscle, okay? If you just see this picture, okay? Cardiac muscle. So let's look at the slide first of all. Let me go out. Zoom out a little bit, okay? So look at this corner piece right here, okay? So on the very outside, you will have the, what layer is this?
Epicardium.
The epicardium, okay? All this is what?
Adipose.
Adipose tissue, pericardial fat is what it is, okay? And on the inside, the layer would be called?
Endocardium.
Endocardium, okay? So what is this part of the heart?
Atrium.
The atrium, okay? And this one would be the?
Ventricle.
Ventricle. So what is this big structure right here?
Artery.
What artery?
Coronary artery.
Coronary artery. Awesome. And what is this structure here?
A valve.
This is a valve, okay? So let's go closer to look at some specialized structures. Who are these?
Purkinje cells.
Yes, who are they?
Conducting fibers.
They're the peacemaker?
Mild conducting.
Conducting.
They're conducting fibers, but they are modified cardiomyocytes is what they are, okay? So Purkinje fibers are right here in the subendocardial layer of the ventricle is where you find these Purkinje fibers, okay? So let's look at another example of cardiac muscle right here, okay? If you zoom in closer, beautiful section. It's just amazing. Okay, so here you can see the fibers, right?
Hey, Dr. Sheba,
Hmm.
could you remember to incorporate just a little bit of time for people whose screens are lagging?
Okay.
Thank
Am
you.
I going too fast again?
I think it's just when you ask the question, then some people like are not seeing the same thing that you're seeing right then, so they need a second, or they just need a second to think before it's getting answered?
Okay, so let me give you some time to think about it when I ask a question. Okay, so here this structure that you see here, you see it, right? What is it?
Circulated disc.
It's the intercalated disc, right? So this is typical of cardiac muscle. I want to point out that you'll see more intercalated discs in this section than in this section. Okay, so if you want to look at intercalated discs, you have to pull this slide out. Okay, so that is that. So now let's go to another piece of tissue right here. Okay, and let's go closer. Look at this muscle. What would you call this?
Still cardiac muscle?
Cardiac?
Think.
It's smooth muscle.
Smooth muscle is what it is. Okay. This is from the uterus. Okay. Smooth muscle. Short fibers, spindle shaped, no striations, and centrally placed nuclei. I mean, I just want you to get an impression of how different smooth muscle can be. Okay. I'm going to show you another example. This is more regularly arranged. Okay. What is this again?
Muscle.
Smooth muscle is what it is. Okay. That is longitudinal section, and this is cross section. Okay. So that is it for muscle. Okay. So we finished the heart, then we are going to blood vessels. Okay. So this big guy right here. Who do you think this is?
There's muscle with a ton of elastic fibers.
An elastic artery.
Elastic artery is what it is. So it could be from the aorta or the pulmonary artery, you know, one of those major blood vessels. And you can see the elastic fibers in them, those wiggly fibers. Okay. You
Hey,
can.
Dr. Sheba, I have a question. On the tunica externa, where we do see all those elastic fibers, like on the outside, where, like, how are those differentiated from the external elastic membrane? Because I see a lot of structures that look similar, whereas like on the tunica interna, we tend to see like the internal elastic membrane alone. So that's easy to point out, but I just wouldn't really know which one to point out as the external elastic membrane in particular.
So the external elastic membrane most of the time is not continuous like the internal elastic membrane is. So it's broken up, you know, doesn't appear as like one continuous elastic lamina. So it's difficult to point out. It will be there in some spaces, it may not be there in some spaces. So you don't really have to worry about it. Okay. But you will see lots of elastic fibers even in the tunica adventitium, you know, in some of the blood vessels. And it's more apparent with trichrome than it is with H&D. Okay. So the thing to remember is it's in these elastic arteries, it's difficult to point out even the internal elastic lamina because there are so many elastic fibers in here. Okay. It's kind of hard. So this is an elastic artery, right? So let's go pick a muscular artery. See, like this guy here, see, there's a beautiful inner elastic lamina, right? Because this is a muscular artery. You can see all the smooth muscle fibers. Very few, there are elastic fibers in here. You can see them like there, but then you see them here also in the tunica adventitia. All these are elastic fibers. Okay, so different parts of the muscular artery. Let's go through them quickly. So that would be the
Tunica intern.
Elastic lamina?
tunica interna, tunica intima. Then this whole thing would be the,
Tunica media.
and this would be the,
Tunica externa or adventitia.
okay, very good. So that is all there is to blood vessels. Then you have to know the different sizes of blood vessels, be able to distinguish like major muscular arteries from arterioles. A capillary would have what? How would you identify a capillary?
Capillaries do not have a tunica media, and they're
Okay.
very tiny, like one red blood cell wide. Tiny.
Okay. So the capillary, like this is an example. Okay, you find lots of capillaries in the lung, right? So we call it a capillary because it has just one endothelial cell lining it. Okay, so here is just one endothelial cell. So this would be a capillary right here. Okay, so there is one capillary there, for example. Okay, just one endothelial cell nucleus. Okay, so we went through capillaries, all the different blood vessels. You have to know like big blood vessels, differences between veins. Veins are here, there is a big vein, but this is a big artery right next to it. Okay, so look at different pictures, be able to identify these different blood vessels. Okay, so heart, blood vessels, we are done. Different types of muscles.
Dr. Sheba,
Yes.
will we be expected to be able to differentiate between capillaries and venules?
No. Like the arterial, like a small vein and venules. So if you have like a structure in comparison, you know, there is a bit small arteriole in comparison. If you see a thin walled vessel right next to it, you can easily point out that it's a venule. Okay, and if it's even thinner wall and just has endothelium, the endothelial layer, you can easily call it a lymphatic vessel. Okay, so we are done with blood vessels. So let's move on to the lymphatic system. Okay, different blood vessels. Do I have any other thing? No. Let's move to lymph, immune. Start with this. No, I should go to immune first and then do immune and okay, wherever I am. No, I should go to the other one. Okay, so what is this section of?
A lymph node?
A lymph node, very good. Okay, so the lymph node, this is not a good lymph node. Let's go to another good lymph node. So here is a lymph node. Okay, so the lymph node has an outer region called the
Cortex.
and an inner region called the
Medulla.
medulla. Okay, so what do we see here? These circular structures, what do we call them?
Nodules.
Nodules or follicles, right? So these nodules or follicles are, there are two types. You would agree? So what are they?
Primary and secondary.
Primary and secondary. So when you look at this nodule right here, you would call it a primary or secondary nodule?
Secondary.
Secondary nodule. Why? Because it has the central region called the
Germinal center.
germinal center and the outer darker region called the
Mantle.
mantle zone. Okay, so the germinal center and the mantle zone. So what type of lymphocytes are in these nodules?
B cells.
B cells. Okay, so the B cells are going to be localized in the nodules, and in between nodules we call that the paracortical zone, right? So that is going to be rich in
T cells.
T cells. So always in any immune organ, whenever there is a follicle, whether it's galt, balt, malt, whatever it is, if you see nodules, it's going to be rich in
B cells.
B cells. And any diffuse lymphatic tissue is going to be rich in
T cells.
T cells. Okay, so that is fundamental. You have to know that. So now let's look at the lymph node quickly. So the lymph node, it has afferent lymph vessels, right? Afferents are the ones that are coming to the lymph node. So afferent lymph vessels are going to come from the region in which the lymph node is situated, okay? And it's going to drain the lymph into these spaces called
Subcapsular space.
subcapsular sinuses, okay? And they are going to move down through the cortex along the trabecular sinus, okay? And it's also going to come down here, right? And then it's going to percolate through the medulla, okay? So when you come into the medulla of the lymph node, you see two regions, right? The darker regions and the lighter regions. So the darker regions are called
Cords.
medullary cords, and the paler regions are called
Medullary sinuses.
medullary sinuses. Okay, so if we go closer into the medulla, we can see a few cells, right? So let's go 40x. So these guys, okay, that have long projections, they're hanging on to each other, you know, and they're forming the stroma of the lymph node. Who are they?
Reticular cells.
They are the reticular cells, okay? The guys with inclusions in them, these brown inclusions, who are they?
Macrophage.
Macrophages.
They are macrophages, okay? And the ones that have this dense dark purple cytoplasm, dark nucleus, okay? These are all these guys.
Plasma cells.
Plasma cells. What do they do?
Make antibodies.
Yes, they produce antibodies.
Could you point those out again, Dr. Sheba? Just like
Okay,
everywhere?
there are a lot of them, but if you look at their nucleus, their cytoplasm is deep purple, okay? And their nucleus looks like a cart wheel, okay? Because the chromatin is kind of pushed to the periphery. Let me find you like a couple. See, look at these guys right here. There is one, there is another one, there is another one. See, there is another one. These are fairly large cells, okay? But they have this marginalized chromatin cartwheel type of appearance for the nucleus. So those are all plasma cells, okay? And there are lymphocytes in general, a whole bunch of lymphocytes in here. So that is that. Macrophages, you have to know when you see the inclusions. So we are done with that. So the lymph flows through the medulla and then it exits through efferent lymph vessels. Okay. Do lymph vessels have valves?
Yes.
Yes, they do. Okay, so now that's the lymph node. So next we'll move on to this structure. What is this tissue?
The spleen?
The spleen. So the spleen has two main regions, right? The blue region, which we call the
Cortex?
no, there is no cortex or medulla on the spleen.
The red.
Red pulp.
White pulp.
White pulp. The blue region is the white pulp. Okay, and all this red stuff filled with red blood cells is the red pulp. Okay, so let's look at the white pulp. So the white pulp is nothing but aggregations of white blood cells, basically lymphocytes, but they aggregate around blood vessels. Okay, so let's look at this blood vessel for instance. Okay, let's look at this guy. See, this is a blood vessel coming in, okay, but there is a sheath of white blood cells surrounding it. So we call it
Pals.
PALS, periarteriolar lymphatic sheath is what it is, okay. So this periarteriolar lymphatic sheath is part of the white pulp, but then there are also nodules like this. See, there is one nodule there, another nodule here, okay. That is also part of the white pulp. So the pals and the nodules make up the white pulp. Okay. So what type of lymphocyte is going to be in the pals?
T cells.
T cells, because it's diffuse, right? And whenever you have nodules, you're going to have
B cells.
B cells. Okay. So that is it. So okay, so let's see. We have this artery in the middle, which is called the
Central artery.
central artery. Okay, so blood is going to flow from the splenic artery, and then it's going to run through trabecular arteries. Okay, and then it's going to split into smaller arteries, and then these are all central arteries. Okay, surrounded by white pulp. Okay, and then it's going to split down even further. It will become peninsular arterioles, probably like these small dots here. Okay, and then it finally enters capillaries, and it flows through this outer zone called the
Marginal zone.
marginal zone. Okay, and then it flows into the sinuses called the
medullary sinuses.
Marginal sinuses. Okay, so there is a marginal zone, and then there are marginal sinuses, and then it continues into the red pulp. Okay, so as these the blood vessels, they become capillaries and they go out here. What happens? The macrophages go and sit on top of these small capillaries, okay, as the blood is getting squeezed out of them and as they flow into these sinuses. So they're going to capture all the dying red blood cells, and they're going to store the iron, because it's in demand commodity, okay, iron, and it's recycled back into the red blood cells. So these are all macrophages that contain hemosiderin. Okay, the pigment is called hemosiderin. You don't have to remember that right now, but it will come in handy when you're studying pathology. Okay, so red pulp, white pulp, pals, nodules, marginal zones, and marginal sinuses. You have to know them in the spleen.
Dr. Sheba?
Yeah.
I know that a few of us were talking after the test, and the blood flow through the spleen was a hard question for us. So do you think that you could review that?
Okay. So it starts with the splenic artery, right? The splenic artery divides and the blood flows through the trabecular artery, and then it divides further and becomes a central artery, right? So that is what you're able to see in cross section. Here is a nice little, and then that divides further and it becomes peninsular arteries, like those right there. Okay, and then the peninsular arterioles become capillaries. Okay, so you find all these capillaries in the marginal zone. Okay, so from there they become sinuses. Okay, capillary sinuses. So here it's like perforated vessel kind of, but then it becomes a discontinuous capillary, technically. That's what we call a sinus, right? So once it becomes that, there is an arterial component and a venous component to these sinuses, okay? So blood flows through the sinuses, through the arterial part, and then it flows into the venous part, and then it runs into the venules, and from there it drains into larger veins that run into the splenic vein, which goes out of the spleen to the caudal vein. Okay, so that is how blood flows through the, but it slows down considerably when it's flowing in through the red pulp, because it's a capillary bed basically. So the blood flow is really slow in the red pulp. Okay, is that clear? Okay, so let's
And could
move
you
on.
explain like how the macrophages get a hold of the red blood cells when they're too old?
Okay, let's go look at it. So this is one section, but this is the bovine spleen. Okay, here let's go closer to look at it. There are lots of smooth muscle fibers. See, all of these are smooth muscle. Have to zoom out a little bit to appreciate the white pulp. Can you see the white pulp? Okay, all these blue areas, dark areas are the white pulp. Okay, so if we go to the marginal zone surrounding the white pulp, let's go closer. Okay. Can you see the structure here? You have to kind of get used to it. See, there is one here, one here, one here. Can you see it?
Tentative, yes.
It's hard to see, but if you keep staring at this image a little bit, you'll be able to distinguish the differences. This is just a bunch of smooth muscle, probably from trabeculae. Here is one. See, there is the capillary, okay? And these are all macrophages surrounding it. So they form these structures called ellipsoids. Okay, so these are the ellipsoids. There is one ellipsoid here, another ellipsoid right here. Okay, just keep looking at it. You will be able to identify a bunch of them. And you can see the ellipsoids only in this section, this spleen section. You won't be able to see it in the other one. So the ellipsoids are nothing but a bunch of macrophages that are sitting on top of these capillaries, waiting for the blood to get squeezed out. And they'll be able to identify all the red blood cells that have reached the end of their life. Okay, because red blood cells are in circulation for about 120 days. So it'll be able to identify those cells that are about to die, and they scavenge them, kind of. They chew them up. Okay?
Are they
And
in
the blood.
I'm so sorry, I didn't mean to interrupt you. Are the ellipsoids in the marginal zone?
They are in the marginal zone and in the red pulp. Okay? Sometimes, see, like here, see they are in the red pulp. They can be present in both spaces. They can be in the marginal zone and in the red pulp. Okay, so let's go back to immune. Okay, so now we are going into diffuse lymphatic tissue. What is this? Yep. Okay, so whenever you see lymphatic tissue like this, see lots of lymphocytes, right? Whenever you see lymphatic tissue with an epithelium, you can happily call it a
Malt? Lymph node?
alt, but it's what? What is it? In ordinary layman terms.
A
A
lymph
what?
node?
I'm so sorry, I didn't hear that.
In layman terms, what is it?
A tonsil?
A tonsil is what it is. So it's associated with the mucosa. Where do you find tonsil? Tonsils.
Your throat?
In the back of your mouth?
So it's associated with the oral cavity. Okay, oral cavity and the pharynx is where you see the tonsils. Okay, so they are right under the mucosa. You have palatine tonsils associated with the soft palate, which is in the back of your hard palate. Okay, the soft palate is an extension of the hard palate. You have to see it in anatomy. Okay, so there is lymphoid tissue buried under the mucosa there. There is lymphoid tissue buried under the mucosa in the pharyngeal area. So there are pharyngeal tonsils and palatine tonsils. We don't have to differentiate them, but you have to know who has crypts and who does not. Okay, so for example, this crypt. So here is a cryptic tonsil. Okay, this has an invagination of the mucosa. Okay, and this is a cryptic tonsil. Okay, while the other one does not have any crypts. Okay, so these are all, both of them are mucosal malts, mucosal associated lymphatic tissue. So here is another one. This is also mucosa associated. So who is this?
Peyer's patches?
Yes. So this is present in what section of the intestine?
The small intestine?
Small intestine, what part?
Ileum.
The ileum. Okay, so ileum is the only area of the small intestine that has the Peyer's patches. Okay, so that is that. Okay, let's go to, do we have one more lymphatic tissue? Yes, we have. One more lymphatic tissue. Here, probably. Yes. Okay, who is this?
Bolt.
This is Bolt, right? Okay, so bronchiolar associated lymphatic tissue. It's not bronchiole, it's a bronchus associated lymphatic tissue because we still have
Cartilaginous plates.
plates, exactly. So this is still a bronchus of some sort, but bronchus associated lymphatic tissue. So here are some more pieces. Okay, so you find this in the respiratory tract. Okay, so now let's move on to, we are done with the lymphatic system. We didn't talk about the thymus.
I was gonna say, did we look at the thymus?
Where is the thymus is my question.
Wouldn't we all like to know?
So this one doesn't have the thymus. Immune and maybe.
Hey, Dr. Sheba, really quick while we're moving. Somebody's asking in the chat, and I don't know how to answer this, with the different types of crypts, or sorry, the different types of tonsils, and some have crypts. Are we expected to know, like palatine tonsils have crypts, or are you wanting us to know the specific species that have crypts?
No. You don't have to know all of that. Just know whether it's cryptic or not.
Okay, so just if there's a presence of a crypt, we should know that that's what it's called. Okay, cool.
Where is the thymus? Cardiovascular, urinary, respiratory, immune. Maybe. Yes. Dang. Okay, so it's hiding here. Let's go closer. Maybe not. Okay, so here is the thymus, right? So the thymus is different from the lymph node. How?
It's separated into different lobes or lobules.
Lobes or lobules. It has an outer region called the
cortex,
inner region called the
medulla.
Medulla. What are the cells that are present in the thymus called?
Is that T cell only?
They are T cells, but they go by a specific name.
Sites.
Thymocytes.
Thymocytes. Very good. Okay, so they're called thymocytes. And in the medullary region, we have these specialized structures, right? Like these guys. Who are they? Like these guys.
Pasals corpuscles.
The Hazel's corpuscle, okay? So this is nothing but epithelial reticular cells converting back to stratified squamous cells. So they are getting keratinized, okay? So that is why you see those whorls in the medulla, especially in older animals, okay? So these are Hazel's corpuscles, typical of the thymus, okay? So we don't have reticular cells here, but we have what are called as
epithelial reticular cells.
Epithelial reticular cells. They are specific to the thymus, okay, to the thymic stroma, and they produce a hormone called
Thymopoietin.
Yes, very good. Okay, so that is it for the thymus. So let's move on to the nervous system quickly. Immune and nerve. Okay, so in the nervous system, okay, here is a section of the cerebellum. So the cerebellum is here, from there to there. This is the brain stem. Okay. So what are these structures called?
Foliate.
They're called folia, right? So if we go look at these folia a little closer, the outer layer is called the
Gray matter?
The outer layer is the gray matter, but it has a specific name, this layer.
Molecular.
The molecular layer, okay? And these cells, these big neurons are called?
Piriform.
Piriform neurons, okay? And this is white matter in the middle, okay? And what else do we have? We have support cells, right? Lots of support cells right here. So there are specific cells, connective tissue cells in the brain. They are called what?
Oligodendrocytes.
In general, what do you call them?
Glial cells.
Glial cells. Okay. So the glial cells are of 3 different types, right? So this guy who is found right around blood vessels, okay, this glial cell is one of the largest glial cells, and it helps to form the blood-brain barrier.
Astrocytes.
Astrocytes, right? Okay. These ones are descendants of monocytes, macrophage family.
Oh, microglial.
Microglia. Okay, so those guys there. See that one there, that one there. Those are all microglia. And then the ones that tend to hang around axons, neuronal cell bodies, that is the
Oligodendrocytes.
oligodendrocyte. Okay, so you have to know the oligodendrocyte is called an oligodendrocyte when it's found in the central nervous system, but when it's in the peripheral nervous system, it's called a
Neurolumocyte.
neurolumocyte or a Schwann cell. Okay, it's what it's called in the periphery. So now let's look at so let's look at this. So this would be a section of the
Spinal cord.
Spinal cord. Okay. So we have to keep it the right way up. So what is the central structure there?
Central canal.
Central canal. So the central canal is going to be lined by specialized cells called
Ependymal.
Ependymal cells. Okay. So all these holes belong to
Myelin.
Myelin. So these are all where axons used to be. Okay. So if you find, so this felt like structure in the background is called
Neuropile.
neuropile. Okay. So all this is broadly, let's say, this would be
White matter.
white matter, and this would be
Gray matter.
gray matter. So if you look at the ependymal cell, so this would be the dorsal horn, right? So if you find neurons in the dorsal horn, they belong to, these cell bodies belong to
Interneurons.
interneurons, okay? And if you find cell bodies down here, it belongs to
Motor neurons.
motor neurons, okay? So now let's look at couple good neurons, and you have to identify parts of a neuron for me, okay? So I can't see this very clearly. Okay, so there is the structure. What is that?
Nucleus.
Nucleus, okay? And if you see a dark structure like that in the nucleus, that would be a
Nucleolus.
nucleolus, okay? So all this blue stuff, what is it called?
Missile substance.
Missile substance, which is nothing but
Ribosomes.
ribosomes. Very good, very good. Hey, I'm liking this. Okay, what is this process?
Dendrite.
Dendrites, okay? But this one would be a
Axon.
axon. Why do you call this an axon?
Because it doesn't contain any missile substance in it.
Very good. So what is this part where the axon takes off?
The axon hillock.
Very good. Hey, this is really good. Okay, so we identify parts of a neuron, right? So similarly, we have to identify parts of a muscle cell. I forgot. Okay. So you have to know what a myofiber is, which is a muscle cell. You have to know what a myofibril is. Okay. What the different parts of the muscle cell. Okay. Like the sarcoplasmic reticulum, what a sarcomere is, and like different parts of the muscle cell basically. Okay, sarcoplasm, and then the actin, troponin, the myosin fibers, muscle filaments. Okay, you have to know all of that, just like you know parts of a neuron. Okay, so parts of the spinal cord, the brain. So when you see bunches of nerve fibers running axons in the central nervous system, like you do right here. Okay, axons in the central nervous system. See, all these are bunches of axons. What do we call them?
Tracks.
Tracks. Okay, so if you find a bunch of axons in the periphery, you would call it a
Nerve.
nerve. Okay, so if you see a cluster of neuronal cell bodies, let me show you one, like right here. Okay, let's go closer. I think, maybe wrong. Oh, these are still active. Oh, here they are. See those neurons?
Mm-hmm.
So if you see a cluster of neuronal cell bodies in the central nervous system, you would call it a
Nuclei.
nucleus. Okay, so there are like specific named nuclei in the central nervous system. Okay, so there is a supraoptic nucleus, the paraventricular nucleus, the Edinger-Westphal nucleus. Okay, so they are all named nuclei. But if you see an aggregation of neurons in the periphery, you call it a
Ganglia.
ganglion. Okay, so let's look at this structure right here. This is what?
Galt.
Galt. Okay, I'm going to go in between these muscle layers. Okay, there are two layers of muscle in the gut. Inner circular and outer longitudinal. Okay, so in between these two muscle layers, let's go closer and see if we can find the structure. There should be like couple. Why is it so difficult? It's there, maybe it's too pale. That is why I can't see it properly. But these are all neuronal structures right here. Okay, so they are called myenteric plexuses. Myo means muscle, so in between muscles, so that's a myenteric plexus. Okay, which is nothing but a ganglionic structure. See, all of these guys. See, that's a good neuron there, but it's colored different, so it's going to kind of throw us off. See, nice looking ganglia. Okay, so in between muscle layers in the gut. Okay, so that's a myenteric plexus. So there is nervous system. So let's go look at this structure right here. See, here is a nice looking ganglion. Okay, aggregation of nerve cell bodies. Okay, and what is this nerve? What kind of nerve is this?
Autonomic.
Do you want to call it autonomic? It's an autonomic ganglion, probably is, but is it myelinated or unmyelinated?
Oh, unmyelinated?
It is unmyelinated, okay? So if you want to look at myelinated nerves, let's go there, cross section. This is not a very pretty nerve, but let's find another one. But this is how it looks in cross-section. I mean, I'm not being very clear. You should be able to see the neurokeratin around the axons. Okay, let's find a better section. There, that one. We go back. Listen to that. Better. So let's go here and take a look at it. Yes, this is prettier. Okay. See, can you see the candy cane appearance?
Mm-hmm.
Still not very clear, but you can see the neurokeratin around the axons. So this is a myelinated nerve in cross section. Okay. If you want to see the same thing in longitudinal section, here it is. Okay, you can see the neurokeratin. So that's a myelinated nerve. So we have another structure here. What is this structure?
A dorsal root ganglia?
Dorsal root ganglia. Very good. So what are these neurons? Are they motor or sensory?
Sensory.
Are they unipolar, multipolar, bipolar?
Unipolar.
Unipolar. Okay, so these are unipolar sensory neurons. And you see them only in the dorsal root ganglia. Okay, so again, if you want to look at myelinated axons, you can see them right here. This is all H and E. So the myelin looks like some kind of a lace wrapping around the axon. Okay, so let's see if we can see it in cross section. No, it's still like that. Okay. That is how it looks, myelinated axons in H and D. Okay. What else do we want to know? So this is another section of the spinal cord. Okay, there is probably a dorsal root ganglion right there. Yes, see, that's another dorsal root ganglion. Okay, so these are all the ganglion, the cells. Okay, these are unipolar cells, and you can see the nucleus, the nucleolus here, neuronal cell bodies actually. Okay. Okay, so let's move on to we're done with the nervous, the respiratory system. Okay, the respiratory system. So here, okay, let's look at this epithelium. So what is this organ right here? Vomeronasal organ. Okay, there are two types of epithelium here. Okay, let's go closer. So what is this epithelium?
Respiratory. Olfactory.
Olfactory epithelium. Okay, so if you go down closer to look at it, all those cells are
Olfactory.
Olfactory neurons. Okay, all of these guys, and the nuclei here belong to sustentacular cells. Okay. Okay, this is olfactory epithelium. Let's go to the larynx. The larynx has different types of cartilages. Okay, so there is, let's take a look at this cartilage here. This would be
Elastic cartilage?
elastic cartilage. Very good. This here would be
Hyaline cartilage.
hyaline cartilage. Okay, and the epithelium here. So this is the epithelium in transition. Okay, so it will be stratified squamous here in the beginning of the larynx, but as you move towards the trachea, you're going to see the epithelium in transition. Okay, so let's look at the trachea itself. So what is present in the tunica muscularis?
A cartilaginous ring.
Yes, you have the cartilaginous ring in the tunica muscularis. Okay, and this muscle, what is this? There is this muscle here connecting the two ends of the cartilaginous ring.
Trachealis muscle.
Trachealis muscle, very good. So the trachealis muscle is a smooth muscle. Okay. So the tunica muscularis contains the cartilaginous ring, and this part alone, it contains this trachealis muscle also. Okay. So there are beautiful epithelium, respiratory epithelium here, and there are some submucosal glands as well. So let's move on to the lung itself. Okay, so this airway is a
bronchus.
That is a
bronchus
still bronchus. Okay, because it still has, that one is also a bronchus, but this guy here is a
bronchiole.
Bronchiole. Okay, so if you go closer, okay, let's go closer some more. It's actually a beautiful example. So here, this would be what we call as the
terminal bronchiole.
Terminal bronchiole. Very good. Okay, and there are some specialized cells in the bronchiole. Let's go closer. Okay, there are ones. Okay, let's look at these guys. Okay, look at these guys. They have a dome shape. Okay, and then there are cells with cilia. Okay, the dome shaped guys don't have cilia, but there are other columnar cells that have cilia. Okay, so there are ciliated bronchial epithelial cells and there are dome shaped ones. So the dome shaped ones are called Clara cells. Very good. Okay, so these Clara cells are present only in bronchioles. Okay, so from the terminal bronchiole, you have the structure. What is this called? This part of the airway?
Respiratory bronchiole.
Respiratory bronchiole, because you have alveoli opening into the bronchiole. Okay, so this would be a respiratory bronchiole, and this passageway would be a
alveolar duct.
Alveolar duct. Okay, that's going down here, and then it opens into an alveolar sac. Okay, so here is probably an alveolar duct kind of branching and going into different alveolar sacs. So here we have an alveolar sac, alveoli, okay, lining it. Okay, so that is an alveolar sac. That is an alveolar sac. Alveolar sac. Okay. So what are in these alveoli? Let's look at the cells in the alveoli themselves. Okay, what are the different types of cells? So there are endothelial cells obviously lining the capillaries, but then there are cells that make up the alveolar walls, right? So who are these guys that are floating freely in there?
Macrophages.
Macrophages. Okay, so these are alveolar macrophages. Okay, and if we have flat cells, squamous cells, they are
Pneumocyte type 1.
type 1. Very good. These are type 1 pneumocytes. Okay, and if we have larger cells like these, that would be
Pneumocyte type 2.
type 2. Okay, so that's a type 2. There's another type 2, type 2, type 1. Okay, so type 1 and type 2 pneumocytes, alveolar macrophages, and endothelial cells lining the capillaries for gas exchange. Okay, you have to know that. So the type 2 pneumocytes produce what?
Surfactant.
Surfactant. Okay, that is essential to keep the surface tension down so the alveolus does not collapse. Okay, the alveolar walls remain patent so air can come in and go. So that is the lung right there for you. So this is another section of the lung, but that one had the best example of the different airways. I'm sorry. Okay, let's move on to the digestive system. Where is the digestive system? Was it? Oh, there, everything digestive. Okay, so we have to look at the tooth right here. Okay, parts of the tooth, you have to tell me. Okay, what is the part from here to here?
Crown.
The crown. From there to there?
Root.
The root of the tooth. Okay, so the outermost part, especially in this section, you can see it way better. Okay, so this part.
Enamel.
So the enamel is the red part here. Okay, so the blue part would be the
dentin.
Dentin. Okay, so the cells that produce the enamel are the
ameloblasts,
and the cells that produce the dentin are the
odontoblasts.
Odontoblasts. Okay, so this is from a young animal. So you're able to see the ameloblasts before the tooth has erupted. So this is an erupted tooth, so you don't see all of that. What is the space in the middle?
Pulp cavity.
Pulp cavity. Okay, so let's go closer to take a look at this. So this is the gum, right? What is this structure here?
Alveolar bone.
Yes, very good. Alveolar bone. And what is this structure here connecting the alveolar bone to the tooth?
Ligament. Periodontal ligament.
The periodontal ligament is very good, very good. Hey, I'm liking it. You guys are ready.
No.
So this part here, what is this?
Cementum.
Cementum, very good. You see it only in the bottom of the root of the tooth, okay? It looks very much like bone and the cells are called
cementocytes.
Cementocytes, and they are present in lacunae just like bone cells. So because they look kind of bone, it's easy for the ligament to kind of attach the structure to the alveolar bone. Okay, so this is the periodontal ligament. Okay, so we are done with that. Now let's look at the tongue. So the tongue has different papillae, right? So they can be either capable of sensing taste, in
Gustatory.
which case we call them
Gustatory.
gustatory, or non-gustatory papillae. Okay, so this one is an example of what?
Phthalate.
This is a foliate papilla.
Foliate.
So all these are foliate papillae, and they have these
Taste buds.
taste buds. Okay, so the taste buds contain special cells called
The chemoreceptor.
gustatory neurons, okay, that are capable of sensing taste, and there is a taste pore, okay, and they also have support cells called sustentacular cells, okay. So this is an example of a foliate papilla. So I'm going to show you another papilla right here. This guy would be a
Phthalate.
circumvallate papilla, okay. So it has a moat around it, and you can still see the taste buds. Okay, so this is a gustatory papilla as well. Okay, so who are these guys?
Glands.
Salivary glands?
Yeah, salivary glands. Okay, so what do they secrete? What is the secretion? Is it going to be a mucus secretion, serous secretion, or mixed secretion?
Serous mucus.
I think this is serous because it has these red granules. Usually when they are red, the secretion is watery. So we call it a serous secretion. Okay. I will show you examples of a mucus gland. Look at this. Okay. Actually, this is a mixed salivary gland because it has these mucus secreting units and it has these serous demilunes around it. Okay. So while we are on the topic of salivary glands, what kind of duct is this?
Striated.
Striated duct. That's very good. Okay. And what kind of duct is this?
Intercalated.
Yeah. It's an absolutely amazing example of an intercalated duct right here. Striated duct right here. Okay. And these guys, look at the connective tissue surrounding them. So they are interlobular. They are in between lobules. Okay, so you can see that the duct is, the epithelium is multiple layered, right? So it's like stratified epithelium right here for these ducts. Okay, so these are interlobular ducts with stratified epithelium. Okay, let's look at another example. Still, this is mixed. It's not pure mucus. No. Okay, this is also a salivary gland. Okay, very good. So if you see the connective tissue surrounding it, then it's in between lobules. So this is an interlobular duct, while these guys are inside the lobule. So these are intralobular. Okay, so that is that. What else do we? Oh, we have to look at the esophagus. So we looked at the foliate papilla, we looked at the circumvallate papilla and the tongue, and then we are going to look at the esophagus right here. Okay, so the esophagus is nothing but a happy tube that goes from the pharynx to the stomach, right? So what type of epithelium is this?
Stratified squamous. Non-cornified.
Non-cornified, awesome. Okay, so here you might see a mixture of smooth and skeletal muscle. You have to be careful here. So let's go closer to take a look at this. Okay, so all of these muscle fibers are in cross section. Okay, but if you look at it, these guys are skeletal muscle. Okay, but this bundle here, can you see it?
Smooth.
That's so here it is smooth. Okay, but this is skeletal, that is skeletal. Okay, so you have to be able to see there is like a bunch of smooth muscle fibers here, but skeletal muscle here. Okay, so different parts of the esophagus, like where it's closer to the mouth and then closer to the stomach, and one part has lots of submucosal glands. Okay, these are all serous or mucus secreting.
Mucus.
Mucus secreting glands. Okay, very good. Look at that. Clearly mucus secreting glands. Okay, so that's the esophagus. Now we are going to move into the this. Okay, you have to tell me what this is.
It's the stomach.
The stomach, the junction of the glandular and the non-glandular portion of the stomach.
Part of the stomach. Very good. Okay, so this is the cardiac region of the stomach. Okay, so there are different types of cells here that we should really look into. So the cardiac portion is right here. This is the initial portion of the stomach. So the cells on the very top, these are called?
Goblet cells?
No. They are present all the way, all around the stomach. They secrete mucus, tons of mucus, but they are called by a different name.
They're called pits.
Mm-hmm. Okay, these are called surface mucus cells. Okay, the pits are these holes. See these holes? So if you look at the stomach surface in gross anatomy, you will see like pinpoint holes in the mucosa. Those are the pits, the gastric pits. They are lined by these surface mucus cells. Okay, and what are these structures? Like when they dip into the mucosa, they form what?
Ducts.
Pardon me?
Ducts.
No, they form glands. Okay, when these structures, the pits kind of extend down into the mucosa and they form glands. We call them gastric glands. All of these are gastric glands. Okay, so the gastric glands are simple tubular coiled glands. So when they come down, they get coiled down at the bottom. So that's why we are able to see them in cross section. Okay, so all these are gastric glands in the mucosal glands. So they have a neck through which they dump the secretion into the lumen of the stomach. Okay, so and that goes out through the pits. Okay, so surface mucus cells right here. This is the neck of the gastric gland. So these are called mucus neck cells. Okay, and then you have the cells of the gastric glands themselves. Okay, so let's go look at what kind of cells. This is not my favorite section to look at gastric glands. Let's move down the stomach. Better piece of the stomach, so you can look at these cells more clearly. Okay, see, same type of surface mucus cells. Okay, here is a nice looking pit coming down. So that's the mucus neck cells, the necks of the gastric glands, and all these are glands covered by these All these are gastric glands. Okay, so there are mainly two types of cells. Look at these cells. These guys, who are they?
Parietal cells.
Parietal cells. Okay, they have these teeny. They look like they are moth-eaten. Okay, they have these holes in them because they are capable of producing what?
Hydrochloric acid.
Yeah, the hydrochloric acid that they produce will like kind of eat that the cell that produces it if it's not shipped out carefully. Okay, so that's why they have canals inside the cytoplasm that when the cell generates the hydrochloric acid, it's quickly moved through these canals and it spits it out right away. Okay, into the these other canals that go out and it gets dumped out through the gastric pits. Okay, so we have one set of cells here. These are the parietal cells. Who are these guys?
Chief cells.
Chief cells. So they produce what?
Pepsinogen.
Pepsinogen. Okay, and it gets converted to pepsin in the presence of hydrochloric acid. And pepsin is capable of doing what?
Protein degradation.
Yeah, digest proteins. Okay, so you want to be very careful with these enzymes and chemicals that the stomach produces, because if it's not careful, it's going to digest itself. Okay, so in order to prevent that, you need a whole bunch of mucus. So that is why we have all these tons of mucus producing cells on the surface. Okay. So chief cells and parietal cells, you have enteroendocrine cells, but we won't worry about that. Okay. What else do we have? So the parietal cells and chief cells, the gastric glands themselves are going to be the thickest and the widest in which part of the stomach?
What was the question? Can you repeat that?
The gastric glands are going to be the longest in which part of the stomach?
The gastric, the middle part, the classic gastric gland.
Which is what?
The glandular stomach.
What is it? I mean, there are three parts to the glandular.
It's not the cardiac and it's not the pyloric.
It's gastric.
It's the fundic part of the stomach.
Fundus?
The fundus. The fundic part of the stomach.
Okay.
Okay. So you have the cardiac region, you have the fundic region, and you have the pyloric region. So the fundic part of the stomach has the longest gastric glands. Okay. So the cardiac part of the stomach is what we saw earlier. See, look at this. It's going to be way shorter. And then when you go towards the intestine, right here, I think. Okay. So here is the small intestine. Can you see it? Let me turn it upside down. There. Okay. So this is the intestine because you're able to see the
villi.
Villi. Very good. Okay. So this is the fundic part of the stomach. Okay. You have to see that it still has the surface mucous cells. Okay. But the gastric glands, you don't see the parietal cells and chief cells anymore. Okay. They look very different. Really not that prominent. Okay, so here is the pyloric end of the stomach. So this would be the beginning of the duodenum, small intestine. Okay, so now let's go to the duodenum. I'm going to pick on, no, I need a better section. Wait, let me see. I'm going to go to this one right here. Let's go closer. Okay, so here is a villus, right? What is this?
Muscle.
Smooth muscle, okay. Where is the smooth muscle coming from?
The lamina muscularis.
No, it's coming from the muscularis mucosa. See, here is the lamina muscularis mucosa. Here is the, what did you say? Lamina muscularis? Yes.
Yeah,
the
she said lamina
muscularis.
muscularis.
Yeah, the lamina muscularis.
Sorry.
I thought we just invented something new that I've never heard of.
Sorry.
I'm to learn something
My
else.
brain is off. So tunica muscularis is down here, but the lamina muscularis is here. So this is like an extension of the lamina muscularis mucosa. Okay. So
I was rethinking everything.
Who is this?
What's what?
Which is what?
This guy.
Submucosal glands.
No.
No.
Intestinal gland?
Intestinal glands, what do we call them?
Mucosal glands?
Mucosal glands, what do we call them? They have a specific name. So these are called crypts of Lieberkühn. Do you remember? The intestinal crypts. Okay. So again, these guys are simple tubular, like coil glands down at the bottom. So that is why you're able to see them in cross section like that. Okay. So the epithelium again is going to be what?
Columnar?
Yeah, simple columnar epithelium is what it is. It's not very clear in this section, but simple columnar epithelium, but you can see the microvilli. Can you see it?
Yeah.
Yep, so the microvilli are here. So this is typical of the small intestine, right? So now let's go to another piece of intestine. What is that? Okay, let's go here. This is another part of my stomach, it looks like. Okay, this is much better. So here you are seeing aggregates of lymphoid tissue, okay, very common in this part of the intestine. But what is missing?
We don't have villi.
Villi, no villi, okay? And these are very simple glands, simple tubular glands. They are not even coiled down at the bottom. The crypts are not coiled, okay? So this would be a typical section of a large intestine, okay? So this is a large intestinal section. Here is another section of small intestine because you're able to see the villi here. So the only difference between the large intestine and the small intestine is the presence of villi. Okay. And here again, if we go in between the muscle layers, you should be able to see look at that, myenteric plexuses. Okay, nerves and neuronal cell bodies in between muscle layers. Okay, so that is that for the gut. Now let's go to the ruminant stomach. So just by looking at these slides here, you should be able to say who is the rumen, who is the reticulum, and who is the omasum. Okay, so I'm going to number this 1, 2, 3. Who is rumen?
3.
3.
3. Okay. Who is reticulum?
1.
Okay. Who is omasum?
2.
2.
Okay. So you have to know the difference in the structure of these different parts. Okay. They're all going to be lined by what epithelium?
Stratified squamous?
Yes. Stratified squamous. That looks a little swollen on the outside because they've been immersed in water, maybe. You know, when they were pulled out of the animal, they would have been wet, and maybe that's why you're seeing this fancy looking squamous epithelium right here, okay? But all you're able to see in the rumen is just these projections. From the ruminal wall, you'll have muscle underneath, okay, the tunica muscularis, and then you have this ruminal papillae is what we call them, okay. So if you go to the reticulum on the other hand, you see these big ridges, okay, and these smaller ones in between, okay. The big ones form the honeycomb. The wall of the honeycomb is what they are formed, okay. But if we go closer to take a look at them, you will see that they have some smooth muscle in their core, okay, which does not connect to the smooth muscle underneath. So isolated pieces of smooth muscle in the core of these big, tall ridges, okay, which form the walls of the honeycomb. But if you go to the omasum, the omasum has these long leaves, right? And if you look in the core of these leaves, you will see smooth muscle. And it'll go and it'll, so let's go there. So it'll go and it'll connect to the smooth muscle underneath. So that is how you differentiate these three different parts of the ruminal stomach. Okay, so that is that. Let's see what that is. That is another piece of the small intestine, but it has submucosal glands. So this is the duodenum. Duodenum is the only part that has these submucosal glands in the small intestine. Okay. The other cool thing is it has these cells called, see, the ones with the red granules. These are called the Paneth cells. Okay, let's not worry about them for the time being, but you should know that there are smooth muscles and there is a structure here in the middle called the, in the middle of the villus, you have what?
Central lacteal?
Yes, the central lacteal. Okay, very important for absorption of nutrients and everything else. Central lacteal. There is a capillary system here actually, okay, which goes to form the portal vessel. So we will come to the portal vessel in just a little bit. So nutrients that are absorbed through these villi, right? They pass through this portal system that starts off as capillaries and then forms the portal vein, and then it breaks up into capillaries again where? You have to imagine this in your head. So it's a portal system is something that starts off as capillaries, becomes a big vessel, and then breaks down into capillaries again. There are only two portal systems in the body. One is this hepatic portal system. Another one is a hypothalamic portal system you don't have to worry about. So this hepatic portal system starts off as capillaries in the intestines from the lacteals, okay? And it takes all the nutrients, flows into the portal vein, and then it comes to the liver, okay? And there it breaks up into smaller vessels again, okay? So that's why we see these portal vessels. Let me go to a good portal area right here. This is from the pig with classic hepatic lobules. So see there, the portal triad, okay? If you go down closer, there's the portal triad. So there is the portal vein, okay? So it's actually the portal vein that comes from the intestines that breaks up into smaller veins, okay? goes to the edges of these hepatic lobules, and then the blood flows from here down through the hepatic sinusoids into the central vein. Okay, from there it flows into the sublobular veins, whatever vein, and then it goes out through the hepatic vein. Okay, so when it's flowing through these sinusoids, the hepatocytes pull nutrients from the blood, or it can pull drugs from the blood, and then it needs to metabolize it. Okay, and then it spits it back into the sinus, or it can throw it into the bile system, into the bile canaliculi. Okay, so the bile canaliculi will take it back down in the opposite direction and dump it into the bile duct. Okay, so there's the bile duct there that's lined by cuboidal epithelium, bile duct. Okay, there is the bile duct. Okay, ultimately it drains into the gallbladder. If it's a horse, what will happen? It will go straight into the intestine because there's no gallbladder. Okay, so that is it. Okay, so blood flows in one direction towards the central vein, bile flows in the opposite direction. Okay, so that is something that you need to remember. So know that this is the classic hepatic lobule. Okay, this is not a very happy looking lobule. Let's go to a happier looking lobule somewhere here. Zoom in. This is congested, this liver, okay? So the sinusoids are filled with blood. This is a happier looking place. So here is a central vein, radiating cords of hepatocytes. And in between the hepatic cords, you have the hepatic sinuses, okay? Let's go closer to take a look at this. So these are the hepatocytes, okay? Central vein. So if we go to the periphery, you should be able to see, hopefully, let's see. Maybe this is not very clear. I wanted to see a happy looking portal triad. Oh, there. See, portal vein, hepatic artery, bile duct. Okay. Okay, so there's another portal triad. So you have to know what liver acinus is. Okay, what a classic hepatic lobule is, what a portal lobule is. Okay, so you have that in your lecture notes, so you better learn. Okay, so here is another structure. What is this?
Gallbladder.
Gallbladder, and it's lined by
Simple columnar.
Simple columnar epithelium. Beautiful simple columnar epithelium. Okay, so these are all examples of salivary glands. Okay, so let's go closer. Okay, what do you think this is? Is this serous, mucus, mixed?
Mixed.
Mixed, very good. Okay, that guy there, who's that?
Intercalated.
Intercalated, awesome. That would be?
Intralobular.
Or they are both are intralobular. They are within the lobule.
Striated.
Striated.
Striated, very good. So this is the striated duct. This is an intercalated duct. Okay, another striated duct here. So we have to move out of this lobule a little bit. Let's see if we can find. an interlobular duct. See, there is an
A ganglion?
yeah, an autonomic ganglion. Okay, I'm looking for an look at this guy. Surrounded by connective tissue, okay? But there is stratified what?
Columnar?
Maybe columnars, maybe it looks like it's stratified columnar. Could be stratified cuboidal or stratified columnar epithelium, okay? And that's present only in the interlobular duct, okay? What is this structure here again?
Got a nerve?
Yeah, this is the nerve ganglion actually. You can see the cell bodies there. Okay, very nice. Okay, now tell me this structure. Okay, you said this is a mixed salivary gland. Okay, this is the what part?
Mucus.
Mucus part. What do you call this?
The serous demilune.
Serous demilune. Very good. Okay, so this would be a mixed salivary gland. Very nice, very nice. I think you guys are ready. Don't want to believe it, but I'm telling you, you guys are ready. Okay, what else did we need to know? Digestive, respiratory, pancreas is here. So the pancreas does not have what kind of duct?
Striated.
Striated duct. It does not have a striated duct. So the intercalated duct in the pancreas starts where?
In the adenomere?
In the adenomere. This is where it starts. In the middle of the adenomere. What is the cell that is present in the middle of the adenomere called?
The shiny, it's called the central
centroacinar cell. Very good. So the centroacinar cell is there, and then you have to look at the pancreas carefully. Okay, so the apical component, apical part of the pancreatic acinar cell is filled with
the acidophilic from the zymogen?
Yeah, zymogen granules, okay? Because the pancreas secretes a variety of enzymes, lipase, amylase, and things like that. So all those are present in these granules. And the basal compartment of the pancreatic cell is blue because of
ribosomes.
Ribosomes. Very good. Okay. So the ribosomes are there. Nucleus is right in the middle, and then you have the zymogen granules on top. Okay. And this structure here would be the
intralobular duct.
Is this a duct?
No, the island, some kind of island, I know it.
Oh, the pancreatic islet, which is the endocrine portion
of the pancreas. Okay. It's called the islet of Langerhans. It has a very interesting story behind it. Okay. It was discovered by one of the guys who discovered this islet of Langerhans. He was a 22-year-old medical student. He was as young as you guys. Made this discovery. Okay, so islet of Langerhans is the endocrine part of the pancreas. So it doesn't have any ducts. So how does it ship its secretions out?
Secretes it into either lymphatics or the blood vessels.
The blood vessels. So if you look at it, see, there are blood vessels right there in the islet. Okay. So what are the hormones that are produced by the islet?
Glucagon.
Insulin, important. Okay. Beta cells produce insulin. Alpha cells produce glucagon. Okay. And delta cells produce somatostatin. Remember just those three. That's good. Okay.
What was the last one you said? I'm so sorry.
Hmm?
Which was the last one you said?
Delta cells produce somatostatin. It inhibits growth hormone. Okay, so this is what?
Is that an elastic artery?
Yeah, it looks like an artery, muscular artery maybe. Yeah, muscular artery. Can you see the internal elastic lamina?
Mm-hmm.
There. Elastic arteries are only present close to the heart. Okay, nowhere else. What else do we have? We are done with the digestive system, respiratory system. I hope I'm not forgetting anything. Connective tissue. I think that's it. Okay, so there are going to be multiple choice questions. You will have some matching questions, some true or false, but I think you guys are ready. You don't want to believe me. You can continue studying, do well on your exam. Good luck.
Dr. Sheba, I love your confidence in me because I don't have any.
No, you know it. You know it. You'll do well.
Thank you so much for taking so much time with us today.
I'm sorry I took this much time, 2.5 hours. Very bad.
I would have been sitting
Thank you,
anyway.
thank
So.
you.
Thank you.
Yep.
Thanks so much, Sheba.
Thank you.
I had one question
Thank
before you go,
you.
Dr. Sheba. Wait.
What?
For when I was going in my review this morning, we talked about the different kinds of glands, or like what they secrete and with what enzymes are we responsible for knowing like what each type of gland secretes and then how like the enzyme that creates that, or just like what they secrete? Does that make any sense?
What glands did we look at?
A lot. It was specifically a section in like the intestinal notes that Dr. J went over, and she like skipped over it, and she said, "We're not really going to look at this." Now we're all kind of paranoid. Like, should we review that section anyway, even though she said we're not going to look at it?
So the intestinal glands, you just have to know that the villi, the crypts, different parts of the stomach, what are the cells that are present there, what they secrete, and then salivary glands, basically, the pancreas, and that's about it.
Okay, cool. Perfect. That was all my questions.
Okay. If you have any other questions, I'm just going to look through this quickly to see if I'm missing something. Just look at all those sections, okay? Just be familiar with it. Identify like different parts. And that's it. You should be ready. You should be good to go.
Thanks, Dr.
If
Sheba.
there's anything that I completely forgot to touch upon, email me.
Right.
Okay, if you have any questions at all, I'll be happy to answer.
Will do. Thanks, Professor.
Okay.
Thank you.
Bye. Good luck, everyone.
Bye.
Thank you.